Glycolysis is a reaction that does not require oxygen, which means that it is an anaerobic reaction. The word glycolysis itself means sugar splitting, as the process involves a cell dividing glucose into smaller sugars. Glycolysis is also a reaction that takes place in a cytosol or the cytoplasm. There are 10 steps in the process of glycolysis. Steps 1 through 5 are what is known as the energy investment stage, but although it is actually using up energy as an investment. Steps 6 through 10 are what is known as the energy payoff stage, as that is when you receive the production of ATP. Step 1 of glycolysis is when a molecule of glucose turns into glucose 6-phosphate after a phosphate group from ATP molecules are added to it. Because of this additional phosphate group, the glucose molecules have a harder time getting out of the cell. The ATP that gave up its phosphate group is then turned into ADP. The glucose 6-phosphate then turns into fructose 6-phosphate, which is an isomer. A phosphate group from another ATP molecule is then added to fructose 6-phosphate to form fructose 1,6-biphosphate. Fructose 1,6-biphosphate then splits to form two 3-carbon sugars, dihydroxacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or G3P. They are isomers of one another, but only G3P can continue through the next steps of glycolysis. DHAP is eventually converted into G3P, as the G3P is constantly being used up during the process of glycolysis. Because of this, G3P will end up being used up before the DHAP is used up. Two half reactions will then occur, as the G3P gets oxidized and NAD+ gets reduced, producing NADH. This is an exergonic reaction, as the energy that is released is used to form 1,3-biphosphoglycerate by adding phosphate groups to the NADH. The 1,3-biphosphoglycerate then gives a phosphate group to ADP in an exergonic reaction, as it releases energy. This causes the 1,3-biphosphoglycerate to turn into 3-phosphoglycerate. The 3-phosphoglycerate is then converted into 2-phosphoglycerate, an isomer. 2-phosphoglycerate then loses a molecule of water, or hydrogen and 2 oxygens, which causes it to turn into an unstable molecule known as PEP that will be ready to lose its phosphate group and has a high potential energy. The PEP then gives its last phosphate group to the ADP, which results in the production of ATP molecules. And because of this, the PEP then turns into pyruvate. And since there are 2 PEP molecules, this is why there are 2 pyruvates, which are the end product of glycolysis.
After the process of glycolysis is the Krebs cycle. The Krebs cycle begins in the matrix of the mitochondria and only occurs with aerobic respiration. In order to start the Krebs cycle, we need to synthesize Acetyl-CoA. In order to synthesize Acetyl-CoA, the 2 pyruvate, which contains 3 carbons, are oxidized so that 1 carbon and 2 NADH are made. This creates Acetyl-CoA, which is a 2-carbon compound. Then Acetyl-CoA combines with oxalic acid, which is made of a 4-carbon compound, to create citric acid, a 6-carbon compound. The citric acid oxidizes, which starts the cycle. This diagram shows the process of the Krebs cycle. The process starts with citric acid. From citric acid to step A, NAD+ is converted into NADH. The same thing occurs from steps A to B. NADH+ turns to NADH by accepting a hydrogen ion and 2 electrons, which will be later important for the electron transport chain.
The end of the cycle, 3 molecules of NADH are created, while 1 molecule of FADH2 is created for each pyruvate. Since 2 pyruvates undergo the cycle, a total of 8 molecules of NADH, 2 from acetyl-CoA synthesis, and 2 molecules of FADH are made, and 6 molecules of carbon dioxide are lost. 2 molecules of ATP are created as well. The creation of NADH and FADH2 are important and leads to the electron transport chain because they will be used to make more ATP. If we take a look at our cellular respiration formula again, we already took care of the glucose from glycolysis. And thanks to the Krebs cycle, we also created 6 molecules of carbon dioxide as waste. and 4 molecules of ATP from the Krebs cycle and glycolysis, leaving 32 molecules of ATP that will be created from the NADH and FADH2 via the electron transport chain. In addition to the 32 ATP, the 6 molecules of oxygen and water remain to be changed. This means that so far, the germinating peas in our cellular respiration lab has used the glucose stored inside them to set up electrons that will yield more ATP in the electron transport chain and also get rid of carbons during the Krebs cycle that will combine with oxygen and be released as carbon dioxide. Also, since the Krebs cycle cannot happen without aerobic respiration, that explains why the germinating seeds used up the most oxygen in the respirometer compared to non-germinating seeds, and the beads because they are not growing and do not need ATP.
NADH and FADH2 are electron carriers that travel from the matrix to the inner membrane where the electron transport chain is located. The electron transport chain uses the electron energy from NADH and FADH2 to pump the protons into the intermembrane space. In the electron transport chain, there are 4 protein complexes embedded in the intermembrane of the mitochondria that transport electrons from NADH and FADH2 to oxygen to form water. In aerobic respiration, the presence of oxygen allows oxidation to occur in these compounds. First, NADH will be oxidized into NAD plus in a complex 1, where the complex will pump 2 hydrogen ions across the inner membrane into the intermembrane space. The 2 electrons from the oxidation of NADH gets transported to an organic molecule called ubiquinone, also known as Q. It moves the electrons to complex 3. Subsequently, FADH2 is oxidized, forming FAD, and its electrons are transported into complex 2. The electrons in complex 2 are passed on directly to Q, which transfers the electrons to complex 3. From complex 3, the electrons from NADH and FADH2 are transported to cytochrome C, an electron carrier that transfers the electrons to complex 4. In complex 4, electrons flow to oxygen. The oxygen will be reduced by gaining the 2 electrons from complex 4 and then bond with hydrogen ions from the matrix to form water. On the other hand, if there isn't enough oxygen to pick up the electrons, then the process of transporting electrons would not occur. As the hydrogen ions enter the intermembrane space, the hydrogen concentration will increase in the intermembrane space. These hydrogen ions create gradient pressure, so ions would diffuse from a high to low concentration area from the intermembrane to the matrix. However, ions are unable to diffuse through the phospholipid bilayer, so they need to transport through facilitated diffusion. In facilitated diffusion, a protein channel called ATP synthase would act as a channel for the hydrogen ions to rush through. When hydrogen ions move through the ATP synthase, ADP would bond with P to form ATP. For every 3 NADH, 3 ATPs are made, and for every FADH2, 2 ATPs are produced. There will be a total of 32 ATPs produced by the end of the electron transport cycle for a glucose molecule, since each private molecule yields 16 ATPs.